Okay, now that we've talked about acid-base catalysis, we can move on, and I'm going to give a general description of the rest of these categories of catalysis. Um, and then next topic, we're going to show a mechanism of protein cleavage that really demonstrates a lot of these other categories in a more tangible sense. So we talked about acid-base catalysis. Let's talk about covalent catalysis. Covalent catalysis is a type of enzyme catalysis where the enzyme actually forms a covalent intermediate with its substrate. So you can think that the enzyme binds to its substrate and then attacks it nucleophilically to make a covalent bond between the enzyme and the substrate. And that covalent intermediate is a stable molecule that sort of stabilizes this transition between the reactants and the products. And that's how the catalysis occurs and why this enzyme substrate intermediate is useful to speed up the reaction. So in order for covalent catalysis to occur, again, the enzyme has to have some nucleophile present in the active site and to form the covalent bond with its substrate. The enzyme has to attack it and form that covalent bond. And then, obviously, the reverse has to happen. In order for that enzyme to restore itself and act as a true catalyst, there has to be an elimination reaction that eliminates the enzyme substrate intermediate, releasing the enzyme to bind to a new substrate. So let's look at sort of a type of covalent catalysis that can occur and try to think of some amino acids that could be in the active site of enzymes that do this. Here's a reaction where we've got acetoacetate being converted over to acetone. And if we look at the mechanism of this reaction, we can see that there's sort of this net flow of electrons from the right side of acetoacetate up to the carbonyl oxygen here. So there needs to be some flow of electrons from one side of this molecule to the other. And then the flow has to go backwards in order to generate the acetone. Now, enzymes can help facilitate that process by reacting with the carbonyl group of acetoacetate. Here, we can think of the enzyme as being this red component here, with the R group not just being a simple organic molecule, but a whole enzyme. That enzyme can have an amino acid that has an amine group available to it. Maybe this is lysine or something. Now, we know that an NH2 has a lone pair and can act as a Lewis base or a nucleophile, and that's what it does, right? You might remember from Orgo 2, if you've taken Orgo 2, that reactions of amines with carbonyls can form things called Schiff's bases or imines. Right? Here's an imine that can be formed by the enzyme attacking the carbon of the carbonyl of acetoacetate. And the thing about this imine is it now has a positive charge on this nitrogen. Now, this is the enzyme substrate covalent intermediate. And this enzyme now has a positive charge on its nitrogen, which really promotes the flow of electrons that we said we needed from this side of the molecule to the other. And so the formation of this enzyme substrate intermediate is promoting this reaction, right? Pulling those electrons into this sink of this positively charged nitrogen. And that allows the carbon dioxide to leave. Then we get a reverse of the reaction where we eliminate the enzyme substrate intermediate, releasing the enzyme to form the acetone product. Okay, so this is an example of how a covalent bond between the enzyme and substrate can cause a flow of electrons that can promote a particular reaction and speed it up. So that's covalent catalysis. Now, our third category of catalysis is called metal ion catalysis. This is very, very common, right? About a third of all known enzymes require some sort of metal, either as an integral part of the enzyme or the metal somehow binds to the enzyme, changes its conformation, and activates it. You've probably heard of some enzymes that require metals. We've already heard of hemoglobin, right, where the iron is, absolutely essential to oxygen binding. But there are other metal metalloenzymes that you may have used, especially if you've taken biology before. Let's talk about magnesium requiring enzymes. A lot of polymerases or restriction enzyme nucleases require magnesium as part of your reaction. And the reason why is because the magnesium shields the negative charge of DNA. We've talked a lot about how DNA is negatively charged because of its phosphate backbone. And if you're an enzyme that needs to bind to DNA because you're going to make new DNA or cut that DNA into pieces, that protein, that enzyme, needs to get close to those negative charges. And that may be difficult, right? There may be repulsion between negative charges on the enzyme and negative charges on the DNA. And so a lot of DNA polymerases, like Taq polymerase that's used in PCR, requires magnesium. The magnesium binds to the enzyme, and the positive charge on that magnesium allows the enzyme to get close to the negative charge of the DNA. Okay? You may have also heard of transcription factors called zinc fingers, right? Those are DNA binding proteins that affect gene expression, and they require zinc. And the positive charge on that zinc allows those transcription factor proteins to nestle in really close to the negatively charged DNA. Okay. Let's show an example of how metals can also be very reactive to ionize water, for example. Here's an enzyme called carbonic anhydrase. This is an enzyme that actually generates bicarbonate in the blood, and it does it from carbon dioxide. Now, how does this work? Well, it turns out in carbonic anhydrase, the enzyme, there's an important histidine, and this histidine works in conjunction with a zinc atom. And these two functionalities end up ionizing water, turning plain water into a hydroxide. Right? So this zinc atom takes water, ionizes it, and holds on to this OH minus, this strongly reactive, very nucleophilic hydroxide. And the metal holding onto this hydroxide then places that nucleophile really close to carbon dioxide to generate our product, our bicarbonate. So in this case, this metal is used to ionize water to generate a good nucleophile that can be very reactive. And so in a similar way that our RNase mechanism worked, we're taking some lousy nucleophile like water, something that's kind of nucleophilic but not very good, and we're reacting it with the enzyme in order to generate a super strong nucleophile, OH minus, and that can promote this reaction. Okay, the fourth type of catalysis that we're going to talk about is electrostatic catalysis. This is really just a concept that a lot of enzymes do, and it is really something that happens when the substrates are bound on the interior of the protein. Now, we said the interior of globular proteins are often really fatty and hydrophobic. We looked at alpha helices and beta sheets and tried to predict where they would be in a globular protein based on whether they have hydrophobic sides or hydrophilic sides. And that was under the assumption that the inside of a globular protein is really kind of fatty. Now, if we bring charges into the interior of a protein, something like a positive charge and a negative charge, those charges are not solvated on the inside of a protein. They're not exposed to water. And we know from intro chem that when we put charges into solution, like water stabilizes those charges. They sort of bind to positive and negative charges, giving them a big fat hug and stabilizing them. And that makes those charges less reactive. And so electrostatic catalysis is really that concept where if you have positive charges and negative charges that are responsible for a reaction, you can make those positive charges and negative charges more powerful, more reactive by excluding them from water, by burying them on the inside of the protein. That makes our nucleophiles and our electrophiles less solvated and more potent so that we get a stronger, quicker reaction. Okay, the fifth type of catalysis is also conceptual, and it's pretty easy to think about. If we look at this slide here, hopefully you recognize this as an SN2 reaction. Right? We've got our substrate here with our leaving group, in this case the X. Our nucleophile is coming in on the back side of our alpha carbon. It's generating a bond as the bond between the alpha carbon and the leaving group leaves, right? This causes an inversion of stereochemistry. You want a strong nucleophile, good leaving group, and you hopefully remember about the SN2 reaction. Now, SN2 reactions occur in the test tube all the time, and you're going to do some this semester in organic lab. But think about how this reaction has to occur. This incoming nucleophile has to attack this alpha carbon in the proper orientation and proximity. It has to attack the backside of that alpha carbon with the correct speed and the correct distance to form this new bond between the alpha carbon and the nucleophile. Excuse me. In a test tube, we're just asking for this orientation to occur pretty much randomly. Our substrate and our nucleophile are just bouncing around randomly in our test tube, and every once in a while, one of the nucleophiles will bounce in the proper proximity and orientation to form this new bond to spit out the leaving group. Now, how can enzymes sort of promote a reaction like an SN2 reaction? Well, we know that enzymes have binding pockets where they bind to their substrates, and they bind to their substrates in very specific ways. And so if an SN2 reaction was occurring in an enzyme catalyzed form, the enzyme would bind to these substrates and sort of line them up perfectly for this new bond to form between the substrate and the nucleophile, and the leaving group could leave. So instead of asking for just random bouncing to allow for this proper orientation to occur, the enzyme holds on to these substrates and sort of places them in the proper proximity and orientation for new bonds to form and old bonds to break. So it's not just random like crazy. You can think of the enzyme as grabbing onto these substrates and sort of placing them together in the proper way. Okay, the last category of catalysis that we're going to talk about is this idea of transition state binding. And this is also called the rack mechanism. Now, this concept was sort of discovered by, looking at proline and how it can switch from the L form stereoisomer to the D form. Now, the mechanism of this wasn't known, but it was thought that these tetrahedral reactants and products go through some trigonal planar transition state. Now, when the enzyme that catalyzed this reaction from L to D proline was analyzed, it was found that there were certain molecules that could inhibit that enzyme. However, the really good inhibitors of the enzyme didn't look like the reactant, they didn't look like the product, they looked like this trigonal planar transition state. And that surprised the researchers. They said, well, the way we think about enzymes is that they sort of bind to their substrates, they undergo the reaction, and they release the products. So the enzyme should be really good at binding to reactants and even products. But why does this enzyme tend to prefer molecules that look like the transition state instead? And that led them to this concept of the rack mechanism, where the enzyme can bind to its reactants, but it really would rather bind to something that looked like the transition state. So what happens is that this enzyme, this racemase, binds to this tetrahedral proline and kind of forces it, racks it into this trigonal planar geometry, which stabilizes that transition state and promotes the reaction. Okay, a rack is, let me look it up. We can Google this thing right now, right? A rack is an ancient torture device, right? Let's get rid of this thing.